Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Christopher Kuzawa. He's professor of anthropology and faculty fellow at the Institute for Policy Research at Northwestern University. He uses principles from anthropology and evolutionary biology to gain insights into the biological and health impacts of human developmental plasticity. A theme of much of his work is the application of principles of developmental plasticity and evolutionary biology to issues of health. And those are the topics we're going to talk about today. So, Dr. Kuzawa, welcome to the show. It's a pleasure to have you on.
Thanks for having me.
Okay, so let's perhaps start by talking about how early life environments impact human health. In this case, since we're talking about humans, of course. So, what are some of the most impactful environmental factors you've studied that play a role in early environments and that have some of the biggest health impacts?
Yeah, that's a great question. So as you mentioned in your introduction, this is an example of what we would call developmental plasticity. And developmental plasticity, you can think of this as the ways that a single genotype, right? So you inherit your genes from your parents. That genotype can generate a range of different traits, biological traits, behaviors, in response to the specifics of the environments that we experience. And this is actually something that my field of anthropology has been studying since the 1960s as sort of an insight into how humans adapt to their environments. So I'm going to give you a quick example to illustrate that. So if we were to fly to, Tibet or the Andes, right, and go to high altitude, we're going to feel that lack of oxygen, that hypoxia. And our bodies will accommodate that initially by increasing our heart rate, and we'll probably get some altitude sickness initially. But over a week or two, our bodies will start to increase production of red blood cells, right, so that our blood can actually carry more oxygen. So that's what we'll do as an adult. But if you were raised at high altitude, your developmental biology will actually work around that stress, okay, by growing bigger lungs. And so the earlier you move to high altitude, the bigger your lungs are. So this is, and if you move, say, as an adolescent, it's really not going to have much of an effect because at that point your lungs are already grown, right? So what this illustrates is the way that development can kind of work around the experiences that we have in the environment as a means of adaptation, but also that there's a critical period when that can happen and outside of which it cannot, right? I'm going to give you one more example, and this will sort of set up an answer to the question that you posed a second ago. Second example is stature. So we think about how tall populations are as adults, and you look around the world, and of course, part of that is genetic, but we now know that most of that variability, when you look at adult populations, right, in body size, most of that traces to nutrition. And when you look at lower resource populations where there's like poor sanitation and a lot of infectious diseases early in life, for instance, during infancy and childhood, those are nutritional stressors that lead to growth faltering. And that stature deficit that is accrued in the first couple years, then some of that carries over into adulthood, right? And so that's another example of a critical period. So the first 2 to 3 years of life, your nutrition has a pretty substantial effect on how tall and how big you're going to end up as an adult, right? So we've been studying those kinds of processes within my field of anthropology since the 1960s. And in the early 90s, an epidemiologist in Britain, David Barker, because he's not an anthropologist, but an epidemiologist, started to publish findings with his colleagues and his collaborators showing that risk for conditions like heart disease, hypertension, diabetes are inversely related to an individual's birth weight. So, we typically think of these conditions as being associated with overnutrition, excess, weight gain, being overweight, etc. This was the opposite, showing that it's actually lower birth weight individuals that go on to have elevated risk for these conditions. And then, animal model studies started to come out showing that if you restrict the nutrition of pregnant mice or sheep or guinea pigs or rats, that the offspring have very similar outcomes, okay, diabetes, etc., as what you see in relation to lower birth weight in humans. So putting all that together, it really started to become clear that prenatal nutrition seems to be having long-term effects on a range of biological traits that influence later health. Now, as an anthropologist, I was in graduate school at the time that these findings were coming out in the sort of early mid-90s. Light bulbs were going off, right? Because this really suggested that there's yet another critical period, right? That's prior to birth, during fetal life, and very interesting in a couple ways. I mean, number one, fetal and embryonic development, there's a lot of development happening, right? So you start with that zygote, the sperm and egg come together, you have a single cell, it divides into two, that divides into four, and so on. And eventually you get all the tissues and organs and systems in the body. But you have to build all of that from scratch. And so there's a lot of potential in those earliest stages of development for environmental inputs and cues and so forth to have large effects. So that's the first thing that was very interesting about this. The second piece is, as I pointed out, anthropologists have been studying plasticity, but it was mostly in relation to things like growth rate or lung volume, these sort of externally measurable, obvious characteristics. This work suggested that aspects of our metabolism, our biology that influence all, these really important disease processes are also governed by these kinds of mechanisms of plasticity. And then the third thing, and I think this is the most, interesting in some ways, is the fact that the environment at this age is, of course, not the environment out there. It's the gestational environment, right? And so it's the mother's body and the mother's biology that's actually sort of influencing fetal development. And all kinds of really interesting, sort of theoretical and ethical implications come out of that, right? So a lot of interest in this stuff in anthropology. I went on to study this for my dissertation. And, we studied it in the Philippines. And like I mentioned, the original findings were David Barker and so on. They were in places like the UK and also in places like Scandinavia where they have really good records, right? These sort of high income countries. There was a lot of interest in how this might play out in a lower income country, because there you have a lot of low birth weight, right? A generation ago or even now. And yet those populations as adults are experiencing, the what we call the nutrition transition, sort of the adoption of high fat foods, more sedentary lifestyle, and an increase in weight gain. And what we've seen is over just a couple generations, what used to be infectious disease as the main sort of causes of death in places like that has transitioned to these kind of chronic diseases. And so the question was, does this kind of Barker hypothesis that I talked about, the low birth weight scenario, play into the rapid rise of metabolic diseases in these settings? And that's what we set out to test, and that's actually, we found very good support for that in the population that I've worked with. So if you're lower birth weight, you see elevated risk for hypertension, diabetes, inflammation, which is related to all sorts of disease processes. And it's low birth weight in combination with gaining weight as an adult that's the highest risk. So it really does suggest that that scenario that was initially documented in the UK could be an important part of why we see very rapid rise of metabolic diseases in lower income countries today.
Right. So let me ask you, is it possible to strictly separate, let's call it normal developmental plasticity from developmental plasticity that leads to pathology?
So, yeah, there's a lot of debate about this. So if you look back at the original, so Barker's original kind of interpretation of what was going on with these findings, it was more of a model of impairment. So I've kind of introduced this as a way of understanding adaptation. But Barker's hypothesis was more about, if a fetus is undernourished, it's going to sort of shortchange some of its organs. It's going to have smaller liver, smaller kidney, et cetera, and this could have sort of negative consequences long term. That's the way he thought about it. Now I think, we have a more nuanced understanding of some of the mechanisms, and we see that there's a lot of complexity involved in some of those processes. There's a lot of epigenetic changes, for instance, that are occurring. And it's pretty clear that it's not all simply about kind of impairing early development. Let me give you an example of that. So if there's a fetus that's undernourished, okay, what you find is that as they put on weight, they put it on the body in a different way than an individual who's higher birth weight. Okay, so it's not that they're prone to put on more weight. As they put it on differently. And the way that they put it on differently actually makes sense in light of the fact that they experienced undernutrition. right? They tend to put it on the visceral depot around the abdomen. And the abdominal fat depot is distinct because there's sympathetic nerve fibers that go from the brain to that depot. And so when you experience a stressor, right, and you feel that adrenaline rush, right, part of what that adrenaline rush is doing is actually mobilizing energy in the body, right? And it's doing that in part through these sympathetic nerve fibers going to that fat depot. So the fact that individuals who experience nutritional stress early in life are preferentially putting fat into a depot that's very rapidly mobilizable later in life, right, suggests that there's something more than just kind of impairment, that there might be an adaptive process. So that's an example, I think, that points to the possibility that some of this is an adaptation of the environment as opposed to simply, stressors kind of negatively impacting or sort of throwing a wrench in the works of early development.
So you talk about undernourishment during fetal development, but what are some of the health consequences associated with, for example, early life malnutrition?
So what are the health consequences? Yes, so I've already alluded to a few of them. A lot of them have to do with metabolic disease because, nutrition is impacting metabolism. Metabolism is sort of how our bodies handle energy, how our bodies handle nutrients, all the chemistry, that sort of keeps our bodies running. That is what is being affected. And those are the underlying processes that lead to conditions like diabetes, heart disease, stroke, hypertension, and so on. Right? So it's really those constellation of kind of chronic metabolic diseases that are the most, the strongest evidence being tied back to early nutrition. But certainly there's effects on the brain, brain development, cognitive development. There's all sorts, as I already mentioned, stature and other outcomes as well. But I would say within this framework, the strongest ties are with things like heart disease, stroke, diabetes.
Mm-hmm. Does developmental plasticity tie in any way with life history from a biological perspective?
Yes, absolutely. So life history is a great topic. It's a little background. When you look across the animal kingdom, you see this incredible diversity of life cycles, right? So you see species like mice that give birth to large litters that they invest very little effort into. Most of them die, right? And then there's a fairly short lifespan, right? And then you compare them to, say, an elephant that grows very slowly, matures very late, which is a big size, has one big offspring that they invest heavily in that's likely to survive, right? And so you've got that whole spectrum. And a basic idea in this field is that variation, you can kind of trace that back fundamentally to patterns of energy allocation within the body. So the body has a finite amount of energy at its disposal. And we know this, like there's a very clear relationship between body size and energy expenditure across species. And a mouse is basically putting all of its resources into reproducing, right? And it's putting very little into sort of maintaining its tissues, slowing the pace of aging, right? So it's a live fast, die young, what we would call a fast life history strategy. The elephant, on the other hand, is the opposite of that, right? It's a slow life history strategy. And we're sort of obviously more on that slow end. So that's what life history is about. That's one perspective on it, is sort of how organisms manage their energy and how that sort of, manifests as differences in patterns of growth and reproduction and so on. Plasticity is a big piece of that because, as individuals, everything I've been talking about so far is arguably demonstrating plasticity in those processes. So as populations experience differences in nutrition, you're going to see that growth rate increases or slows down, as a way to accommodate that, and everything cascades from that. If you're growing slower, then your maturational rate is going to be slower, et cetera, right? So that whole process of the human life history and how it's evolved is also very plastic and kind of can work around our experiences, this kind of the specifics of our local realities.
Mm-hmm. To understand how our life history as humans evolved, is it also important to understand aspects of the evolution of our brains?
Absolutely. Yeah. So the brain, anthropologists have been obsessed with brains for a long time, and with obvious reason, because It is the organ that most sets us apart from other species. It underlies our cultural complexity, our technology, our language, etc., right? And so it clearly is important. And from the perspective of everything we've been talking about, it's super important because our brains are very energetically costly. And so this has been a big piece of the interest among anthropologists through the years. The classic figure is that the brain in the human accounts for about 20% of our energy expenditure, which is true, okay? And it doesn't necessarily seem like that much, but if you compare us to other mammals, that figure might typically be something more like 3%, right? So it's much higher in us. And so there's been a lot of interest in how do we actually afford that? What are some of the other functions that might have been shortchanged in order to subsidize that? Well, my interest since graduate school has been thinking about how those costs play out and how we manage to afford the costs of our brains during development. Because, just think of a baby, right? The size of the baby's brain relative to its body is enormous, right? It's all head. And so that 20% figure is obviously going to be a lot bigger early in life. The challenge, however, is finding a way to measure that. How do you actually measure how costly the brain is? And you can't just measure, the cost of the brain in the adult and make sort of assumptions, extrapolate that to the kid, to children and infants, because the cost of the brain actually varies a lot because of the extra costs associated with learning. And this was shown really elegantly back in the 80s by Harry Chugani, who has this unique data set that he collected when he was at UCLA, and this is PET imaging data, so positron emission tomography. The principle of PET imaging is that you give somebody a glucose that has a tracer, and then they sit in a scanner, and then you can actually measure the rate of glucose uptake by tissues, right? So you can actually quantify energy use in the brain. And kids that were referred to him that ended up being healthy, he got their permission to put their data into this data set, and so he has this unique data set that goes from birth until adulthood. And what these data show is that each gram of brain in a child around 4 or 5, 6 years of age is using twice the energy of the adult brain, right? And what this traces back to, as I alluded to, is the cost of learning, right? So one of the ways that we learn, that the human brain learns, is that we generate excess neuronal connections, synapses, during development. And then those pathways that are used, as we use our arms and we interact with the world, we're firing those pathways. Those that are used are stabilized. Those that are not are pruned away, right? And so eventually we sort of prune things down to the adult level and get at that 20% of energy expenditure going to the brain, but not before we go through this period where there's this kind of peak in energy being expended related to learning. So what we did is I collaborated with Harry Chugani and others to bring those data together with MRI data, right? So the PET data tell you how much glucose each gram of brain is using. We then need to know how many grams of those different structures there are, right? So use the MRI data to do that, multiply that all together to get the total cost of the brain. What we find is that the cost of the brain is peaking out in childhood, when those synaptic processes are highest, between 4 and 5 years of age. And the 5-year-old's brain is actually using twice the total energy of the adult brain, which is kind of remarkable because the size of the body of a 5-year-old is 1/3 the size, of the adult's body, right? So you can start to see that this is a huge kind of energetic drain on the body. And so we quantified that, and we found that fully 2/3 of the body's resting energy is going to the brain between 4 to 5 years of age. That's the peak, in the human life cycle. So 2/3 of calories, just of a 5-year-old who's sitting there, are going just to keep its brain going. And so you can imagine how this is going to have major implications for the life history, right? This is going to kind of distort the life history because it's going to end up sucking resources away from other aspects of biology, and we found very strong evidence for that. So there was an idea back, again, in the anthropological literature, if you go back, folks had hypothesized that one of the reasons that humans grow so slowly, so if you look at our children, human growth is extremely slow compared to other species and other mammals, and it had been hypothesized that this might be sort of a compensation for the extra cost of the brain, right? So we're kind of diverting resources from growth to the brain. And we tested that with these data, and we found very strong support for this. So if you look at growth rate and the cost of the brain, they're tightly inversely related from infancy until puberty. And it's just almost a perfect linear relationship. It was much cleaner than any of us would have anticipated, actually. So what this shows is that as you know, our brains are so costly that we've had to kind of divert resources from other aspects of our biology, especially our growth rate, in order to subsidize this. And so, you know, our life history has been shaped by this. You know, we have very slow growth rate during childhood. Presumably that's going to require an extension of the period of maturation as well, right, to reach the same body size, right? So it's actually the brain and its energetic cost is a big part of kind of, you know, we need to understand those costs to understand other aspects of our life history and how they evolved, especially this very slow growth rate.
Right. Are there any major individual differences in developmental plasticity? So, to be more concrete, is it that different individuals have different levels of developmental plasticity? Some of them are more plastic than others, or could it be also that, for example, individuals are equally plastic but in different directions? So, for example, some of them would respond to the same environmental factors, environmental stressors in different ways.
Yeah, that's a good question. And I think it's understudied, honestly. I think we know more about species differences, you know, in plasticity. We know more about differences in, you know, like I was talking about with critical periods, right? So there are certain periods of the life cycle that are more sensitive than others, and that have effects that are, you know, specific to certain outcomes. But, you know, there's an assumption that there are going to be differences in plasticity across individuals, but it's sort of understudied. And yeah, so I you know, I don't think we know a ton about that. Certainly there's going to be, you know, there's going to be, you know, the basic assumption is that our capacity for plasticity is based in our genes, right? So part of why we're plastic is that our genes code for flexible systems. But the parameters of those flexible systems and the sort of ranges of possibility within them are presumably variable based upon, you know, individuals' genotype. But again, I don't think we know enough about that at this point. It's been understudied.
Yeah, fair enough. So you mentioned that you've done some studies in the Philippines, and I have here that you've studied people in Cebu or Cebu, I'm not sure how it's read,
Cebu. Yeah.
for more than 3 decades. So could you tell us what are the kinds of insights that you get from studying people for that long, 3 decades in that case? I mean, the sort of long-term studies that you wouldn't get from more short-term studies.
Yeah, sure. So yeah, I've been very fortunate to be able to collaborate with this study in the Philippines. And this goes back to sort of the topics that we were talking about at the beginning. You know, how do you, what are the effects of early environments and how do they influence later health? That has been a big focus of what we've done in the Philippines. A little bit of background on that study. I mean, it started in 1983, before I was involved in it. It was designed by economists to look at the effects of formula feeding versus breastfeeding on infant growth and survival. So this was back when formula companies were promoting the use of formula in lower resource settings and poorer countries as a sort of more modern and preferable way to raise kids. And we now know that that's not true. And part of the reason that we know it's not true is because of studies like this that were actually designed to look at that. You know, it basically showed that infants that were breastfed were protected. You know, they grew better and they had better survival. So that was the initial focus of the study. And to do that, they enrolled more than 3,000 women when they were pregnant, and then they tracked the kids every 2 months, you know, for 2 years. So it was a very big effort, and it was representative of a whole metropolitan area, the Cebu metropolitan area that you talked about. But now, you know, actually, then when the kids were about 8.5 years of age, they found money to go find them, you know, to relocate them, and then turn the study into a longitudinal study. So you can start to track them and look at things like how they grow. And so the questions have changed, you know, as the population has aged. I got involved in the study when they were sort of in adolescence because of my interest in this Barker hypothesis, right? The stuff that we talked about, the idea that undernutrition early in life might set up risk for increased, burden of metabolic disease later. And so we started to look at those questions, in the late 90s. And now that original cohort is almost 40 years old, right? So the study is still tracking the original mothers who are quite elderly at this point. And then you've got the birth cohort, who are in utero when the study began, that are now almost 40. And then you've got their kids, right? So this is a pretty remarkable resource for trying to understand how some of these lifelong processes influence health, how some of these processes might have intergenerational components, and specifically how this all plays out in a population in the Philippines, which is more typical of many of the world's populations, kind of, lower middle income country, urban setting, right, with in that kind of context. And I already talked about some of the things that we found. We find that lower birth weight and measures of poor nutrition early in life set individuals on, a path to having higher burden of metabolic disease later. We've also had a big focus in this study on immune function and immune development. Immune regulation has been a piece of that. And then as the population has aged, the other opportunities to look at other kinds of questions have also emerged, right? So as the original cohort became parents, we started to look at questions related to parenting and reproductive biology and so forth in both men and women. And so one focus of that, which is completely unrelated to what we've talked about so far in terms of plasticity and early environments and the long-term effects, is looking at the hormonal changes that fathers experience, or as men become fathers. And this is a really fascinating story because, most mammals, let's back up a little bit and do a teeny bit of evolutionary biology background here. Most mammals, the father doesn't even know which offspring is his, right? And so they're completely checked out. There's no way for them to even be, to invest in the offspring. But in birds, in like 80% of birds actually have biparental care, where both the male and the female are involved in raising the offspring. And there's good reason for that, right? Because you've got to incubate. There's a lot of things that have to be done to successfully rear birds to adulthood, right? You've got to lay the eggs, you've got to incubate the eggs, you got to protect the nest, you got to feed the chicks, etc. So it actually many times takes both parents to successfully do that. And so what you see in these species is that testosterone in males tends to be very high during the mating season, right? When they're sort of competing and guarding territory or whatever it is for that particular species. But then once they become fathers, their testosterone plummets, right? And the idea is that, or the sort of hypothesis is that this is kind of helps them reorient their behavior around nurturing, and sort of keeping, minimizing the sort of competitive tendencies. And researchers have shown very elegantly that this is really what is happening, because they've given testosterone patches to male fathers, and they leave the nest and go kind of defend territory and fight and stop feeding the chicks, right? So it's pretty clear that it's adaptive to downregulate your testosterone as a way to sort of reorient your behavior. Now, humans, of course, are more like birds than most mammals. Like we do, typically know as fathers, we tend to know who our child is because we have relatively stable pair bonding, and then we tend to be involved, right? We have biparental care. So some decades ago, anthropologists started to ask, like, do we see evidence for this kind of decline in testosterone in humans as you see in birds? Do you see something similar? And they had shown, mostly just kind of what we would say cross-sectionally. So you go into a room or you go into a classroom and you have everybody spit in a tube and you ask them whether they're a father or not, whether they're in a stable relationship or not, and then you just sort of tabulate things. And what they found is that the guys who were not in a relationship and were not fathers had the highest testosterone. The ones who were in a relationship but were not fathers had sort of lower, and then the lowest were the guys who were in sort of married, fathers, right? And so that was consistent with the idea, that the bird model might, sort of be analogous to what we see in humans, but it was cross-sectional, so it wasn't clear what was kind of cart and what was horse, right? One possibility is that as men become fathers, their testosterone declines as a way to sort of reorient their behavior. But the other possibility is that guys with low testosterone to begin with are more likely to stick around and become fathers, right? So it could go either way. And so we set out to test that longitudinally, like looking at the same men, before they were fathers and then after, using the data from the Cebu study. And what we found was very strong support for the kind of bird model, right? So among guys who were not in relationships and were not fathers at baseline, the ones that had higher testosterone to begin with were more likely to become fathers, okay, by follow-up about 4.5 years later, right? So having high testosterone when you're a single guy in this population seems to be predictive of actually ending up in a relationship later, right? But once they did, their testosterone plummeted below everybody else, right? So testosterone was sort of encouraging mating and entering these relationships, but then the act of becoming a father seemed to really drop testosterone. And the amount of time that men spent in direct care was directly inversely related to their testosterone levels. If they co-sleep on the same bed surface, okay, there's a lot of co-sleeping in the Philippines, their testosterone is lower, right? So there's something about the act of, being an involved, engaged father that seems to drive down our testosterone levels. It's really fascinating because, again, it's not something that you see in other mammals. Typically, I mean, there's some examples, but they're kind of, they're sort of unusual. And it really suggests that males, and the role that males play in raising kids, has been important enough to our kind of success, reproductive success of the human strategy, that it's kind of, evolution has rewired our biology, right, to sort of converge with what you see in birds. So it's a really cool story.
Yeah, it's really cool. So now for the second part of the interview, I would like to ask you about epigenetics, specifically human epigenetics. So could you tell us to what extent is human epigenetics developed? I mean, is it in its early stages? Is it well developed? Do we know many epigenetic mechanisms that operate in humans, for example?
Yeah, first of all, I'm not a molecular biologist. I am not, I do not have primary expertise in this. I mean, I do collaborate. We do measure epigenetic markings in our data, and we do analyze them in collaboration with others. And it's something that I'm really interested in. But I'm not necessarily an expert per se, but I'll give you my take on it.
Okay.
I think that, first of all, what are epigenetics? For listeners who might not be familiar with this concept, you inherit genes at birth, right? You inherit certain genotypes, which is the nucleotides on your DNA, your chromosomes. And there's been this kind of growing awareness that part of what matters for your biology isn't just what genes you inherit, but it's which genes are actually turned on and where, right? So every cell in your body, for the most part, has the same DNA. So what differentiates a liver cell from a neuron or whatever is not which genes are present, it's which genes are actually being turned on and off, right? And so those processes of sort of regulating which genes are being expressed and where is what epigenetics is about. It's about sort of chemical modifications to the DNA that can silence genes or increase their expression. And so some of the processes that are best studied in humans, one of them is methylation. And this is partly, I think, a function of what's easier to study. But methylation involves the attachment of these a methyl group at particular regions in the DNA. Typically, this, if a gene is more highly methylated, it's going to be silenced. And so, that's the sort of work that's been done. And there are now chips where you can measure methyl marks across close to 1 million sort of sites across the DNA. And that's the sort of way that a lot of this work is being done, right? So you measure methylation across the genome in a lot of people, and then just like with genetic research, now you're saying, you're looking to see if methylation at specific genes predicts disease or, is related to different kinds of behaviors or outcomes. I think there's no question that epigenetic processes are important in humans. I mean, they're important in all multicellular organisms because it's integral to the process of development. Like as I was just saying, the way that a neuron becomes a neuron and a liver cell becomes a liver cell is through epigenetic processes, right? What's new over the last couple decades is the realization that those processes are actually sensitive to the environment, right? So this kind of ties in with some of the things that we talked about early, how early environments shape later health. Well, some of the ways that occurs is through changes in these methyl marks, or changes in different kinds of epigenetic changes that then persist into later life to influence health. And I think, I should say that there's sort of two big domains in which this occurs, right? So most of when you hear about epigenetic research and the kinds of stuff that's being studied in humans, almost all of that is focused on what we call somatic somatic inheritance, right? So these are epigenetic changes that are occurring in a tissue and that persist in that tissue, all right? And once we die and our bodies go away, those epigenetic marks go with it, right? So they die with our bodies. They're not necessarily transferred to the next generation, right? So it's really a process of a set of pathways that link experiences to later health, right, in the same generation. That gets interesting, of course, when we start to think about the fetal environment and epigenetic sensitivity there, because now epigenetic marks can be influenced by the mother's experiences, by hormones coming from the mother, and so on. So there are interesting intergenerational possibilities that are not necessarily the inheritance of the marks themselves, but sort of these intergenerational effects that are influencing those marks in the next generation. So that's all about the somatic cells. The other domain, and it's more contentious, and it's, I think, in some ways really, really fascinating, has to do with epigenetic marks that are passed on to the next generation through the germ cells, through sperm or egg. Right. And this is now, I think, very convincingly demonstrated in other species. So let me give you some examples of this. So in mice, a lot of this work is done in like rodents, right? And this is an area where a lot of the work has been done focusing on paternal, that is male experience, because there's a challenge when you're looking at these kinds of effects of ruling out other sort of confounding explanations, right? So if you stress a female, right, and then you kind of look at the sort of biological changes in the next generation, it could be that there's some sort of epigenetic change going through the egg, or it could be that the gestational environment is different, or that the mother raises has differences in behavior and raises the offspring differently, or whatever, right? There are other kinds of pathways that operate, or lactation is another, right? There's signals and so on that come through breast milk. In fathers, you can really isolate, you just take sperm and sort of, isolate it and keep the father out of the picture, right? And this is a way of kind of really trying to hone in and see if the effect is going through the germline. And this work is really pretty fascinating. So there are, some of the interesting examples are things like, if you change the diet of a father, and then you cross foster and so forth with mothers who are exposed and whatnot, you see that cholesterol levels are different in the offspring, okay? And it's clearly coming from the sperm. There are effects on stress physiology. So if a father is stressed out, and then, like there are protocols for stressing out rodents, their offspring will actually kind of produce stress hormones in a different way and have different reactivity to stress. In those cases, they've isolated specific epigenetic factors in sperm and isolated them, pulled them out of sperm, injected them into the sperm of unexposed animals, and replicated the same phenotypes, really. And so very kind of definitively demonstrating, in my view, that there's this kind of inheritance process happening. That literature is focusing not so much on methylation, but on non-coding RNA. So most of our genome, there's something like 3% of it that actually codes for proteins, and the vast majority of it is non-coding. It doesn't actually make it to the protein phase, but it actually has a lot of effects, right? So it has these transcripts can regulate production of other other proteins and so forth, and it's those non-coding non-coding RNA transcripts that seem to be modified by our experiences and that are transported down to the sperm, packaged up in the sperm, passed on to the next generation, and influence health and biology. So very, very well demonstrated in mice, I think. We do not have clear, definitive demonstration of this in humans yet. I don't think there's any reason to suspect that our biology is fundamentally different than these other species. I think that we are going to see, if you come back, if we can rewind, sort of fast forward 10 years into the future, I think we'll probably have pretty good evidence for this kind of thing in humans. How important will it be? I don't know, but it's really fascinating. And one of the reasons that it's fascinating, the research that I was talking about earlier is all, focused a lot of attention on mothers, right? And the sort of gestational environment and the fetal development and as having this kind of fateful influence on later health. This other research, I think, is really showing that male experiences, male behavior, can also have intergenerational effects, and it's going to bring males into that intergenerational story.
Right. So how do you think about the relationship between epigenetics or epigenetic mechanisms and the sort of mechanisms that we find associated with the modern evolutionary synthesis in evolutionary biology?
Yeah. So, right. So there's a big movement right now within evolutionary biology to kind of look back at the history of the field and think about whether the foundations that were laid, when Mendel's, the rediscovery of Mendel's insights into genetic inheritance, when that was brought together with Darwin and Wallace's insights about natural selection as the sort of engine of genetic adaptation, that was a process, that was a historical event called the modern synthesis. It lasted from, say, 1930 to maybe like around 1950, right? It was the bringing together of those two frameworks, and that laid the foundation for modern evolutionary biology. What you see now is that there's kind of, there's a reckoning and trying to think about how do we fit in some of these ideas about plasticity, about epigenetic sensitivity, about alternate pathways of inheritance, right? That there are, we have more and more evidence that there are kind of ways of inheriting biological information that are not, limited to nucleotides. Some of it might be methylation or non-coding RNA or cultural systems or what have you, right? And that is called the extended evolutionary synthesis. That's what some have called it, right? It's this sort of trying to integrate these newer ideas into the framework of evolutionary biology. And one of the areas that I think is particularly interesting, and that where some of the stuff that I've been working on, I think interfaces with it, is thinking about plasticity, and epigenetics is part of that, right? This kind of sensitivity to the environment, how that plays into the evolutionary process. Because the old assumption, like if you think about how most of us learned about genes and how they work, right? If you think about old textbooks and probably even modern textbooks, I haven't looked at like what high school students are learning today, but the examples that we always learned were Mendelian traits, right? Like eye color, so if you inherit, 2 blue eye genes or whatever you want to call them, alleles, then you're going to have blue eyes, right? And so it's this very simple kind of system, and it's akin to what Mendel discovered with his peas, right? With wrinkled peas and yellow pea pods and so forth. The problem with that is that most traits don't actually work that way. They don't work that simply. The pattern of inheritance is a lot more complicated than that. And, the genome doesn't just kind of simply determine the trait. Instead, what happens is you have these flexible systems, as I've been talking about. You go into an environment, and the environment actually is the sort of source of the phenotypic change, typically, and the initial change that occurs. So I talked about the example of lung volume. You move to high altitude, you don't have to wait around for many generations for some mutation to, arise that has some little small effect on lungs that you then pass on to your kids and grandkids and slowly takes off in the population. You can just grow bigger lungs because you have these developmental processes that allow that to happen in real time in response to your own experiences, right? So what that means is that the origins of phenotypic novelty tend to be plasticity in the environment shaping plasticity. Another example of this, so there's a wonderful book that lays out a lot of these arguments by Mary Jane West Eberhard. It's kind of a classic already. It's only less than 10 years old, but I'd say it's a classic, Developmental Plasticity and Evolution. And she gives this wonderful example in there of the two-legged goat. So there's a goat that was born congenitally without front limbs, and it had to walk on its hind limbs. It basically became habitually bipedal. What you see is that there's a complete reorientation of its skeletal system, right? So its pelvis is reoriented, the muscle attachments are all reoriented, the robustness of the bones are all changed, and so forth. And that's because of plasticity, right? So if you think about like the evolution of bipedalism in humans or something, right? I think it's a really interesting sort of insight into that. The driver of that process was no doubt behavioral, right? So maybe the environment was changing, it was becoming more arid, maybe it was becoming more open in savanna. And so behaviorally, early hominids, quadrupedal ancestors to hominids would have started walking bipedally, kind of in a clumsy way, but that would actually reorient the skeletal system, right? And if you start doing that at a young age, it's going to reorient it more. That adaptation to bipedalism is going to be behaviorally driven first, and then the genome is only going to slowly and later catch up to that. Another example is, we've talked about high altitude. If you look at populations that have been living at high altitude for long periods, many thousands of years in Tibet, in Nepal, in the Andes, and Ethiopia, what you see is that there's been genetic adaptation that in all those populations that allow them to inhabit those high altitude environments. But what initially allowed them to get there, was the stuff that I was talking about, the developmental responses and so forth, and the increases in red blood cell count and all that. All those kind of phenotypic and developmental responses is what allowed migration into those high elevation environments. And then only gradually, did the genome catch up to that. So the model that comes out of this is one in which the phenotype is the driver, really. It's the responsiveness of the phenotype that's generating the variants, the phenotypic variants that are then selected through by natural selection. And then again, the genome is the caboose. It's kind of bringing up the rear. Epigenetics, I think, is part of that, right? Because that is part of how we have plasticity, these kinds of these mechanisms that we've been talking about. Epigenetic processes are integral to that. And then there are also the sort of germline processes that really get interesting, but that's a whole nother story, I think.
Sure. So you wouldn't say, I guess, that, for example, extending the evolutionary synthesis to include things like epigenetics would trump what we already know in the modern evolutionary synthesis. It would simply complement it. Correct?
Yeah, absolutely. I mean, I don't think there's anybody. I shouldn't say that. There are very few people that would question, the importance of natural selection operating on gene frequencies. That's obviously really important, and the genome is the kind of stable, very high fidelity, heritable component that provides that kind of ratchet, right? So as you generate as you generate novel phenotypes and they're advantageous and they improve survival and reproduction, it's the stability and the high fidelity of the genome that actually allows that to kind of stay in place and ratchet up increasing complexity. So that's unquestionably an important part of the story. It's just an incomplete story is all. We just realize that there are these other processes involved now. And what they involve, I mean, there's other elements of the extended evolutionary synthesis. Another is, niche construction. That is, that's another really interesting idea, right? So, in classic evolutionary sort of neo-Darwinian theory, the environment is kind of selecting for traits that are genetically based, right? And that's the basis of natural selection. Those traits that are better fit become more common because the carriers are more likely to survive and reproduce, right? So it becomes more common through time. The environment is this thing that's out there, and it's just sort of like is the filter of the whole process. But it's common sense that most organisms are not just sort of experiencing this environment passively, but they're kind of actively constructing their environments, right? And so, the classic examples of that are things like are species like beavers, that create this dam and actually completely transform their ecology. What this means is that the environment is actually partly a product of the genome, right? So it sets in motion some feedbacks. And, these kinds of feedbacks can take on a life of their own, really. And you cannot sort of simply boil that down to, natural selection operating on the genome. Like there are dynamics to those kinds of processes that are independent of that, much as there are with the inheritance of epigenetic information across generations and so on. So it doesn't overturn the idea that natural selection operating on genes is important. It just says that there's a lot more going on. Some of it might be faster timescale. Some of it might have sort of reciprocal effects where the organism is both creating and being affected by the environment in a bidirectional way. There's just a lot more complexity than that original model allowed.
Yeah. Okay, so just one last question. Since we have genetic adaptations, do you think that we can also talk about epigenetic adaptations? I mean, is there any of that?
Epigenetic adaptations. Yeah, I think so. It's hard to establish that, definitively, right? Because to really show that an epigenetic change is adaptive, you need to be able to link it up to survival or reproduction. The vast majority of research on epigenetics is motivated by different goals, right? It's to demonstrate or to kind of uncover the role of those processes in health and disease, which are not necessarily the same thing, because the causes of health in a modern environment where we're surrounded by high-fat foods and chronic stress and so forth, it's not really giving us insights into our evolution per se. So it's hard to definitively establish this, but I do think, and I've sort of already alluded to some of this, I think that we can kind of infer that some processes are likely adaptive based upon various criteria. And one of those is complexity. Like when you see evidence that changes are kind of implausible, to be just by chance, and where you have multiple systems that are sort of working together to create a phenotypic change that makes sense in light of the inducing stimuli, right? So if you have undernutrition early in life, and then you see the body kind of setting up, through epigenetic and other kinds of mechanisms, a change in how it responds to undernutrition in ways that seem adaptive or sort of helpful, then I think that would, that is sort of de facto evidence in support of this idea. But I think that's kind of where we're at. I don't know that we have definitive evidence at this point, but I think it's extremely likely that, just as developmental plasticity is incredibly important as a means of adaptation, the epigenetic contributions to those processes are obviously an important part of that story as well.
Okay, great. So just before we go, Dr. Kuzawa, would you like to tell people where they can find your work on the internet?
Oh, sure. Yeah. I have a web page, but it's not very updated. I think my Scholar page is the way to go. Yeah. And pretty much everything that we've touched on today, you'll be able to find there. So hopefully that's...
Okay, great. I will be leaving links to that in the description box of the interview, and it's been a real pleasure to talk to you. Thank you for coming on the show.
Thank you. It's been great. Appreciate it.
Hi guys, thank you for watching this interview until the end. If you like what I'm doing, please do not forget to support the channel. You can go over to Patreon at patreon.com/the Dissenter. And you also have links to PayPal in the description box of the interview. Otherwise, and if you like what I'm doing, please share the interview, leave a like, leave a comment, and hit the subscription button. This show is brought to you by Enlights, Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and supporters, Karen Litzke and Blanchett, Paraga Larsen, Lau Guerrero, Francis Fordens, Frederic Sunda, Ricardo Vladimiro, Craig Healy, Adam Castle, Olaf Alex, Jonathan Wissel, Jacob Klinkby, Matthew Whittingbird, Arno Wolf, Tim Hollacy, Eric Alenia, John Connors, Paulina Barron, Philip Forrest Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingard, Rebecca Newberger Goldstein, Dan Demetriou, Robert Windager, Rui Inácio, Arthur Coe, Zoop, Marco Neves, Colin Holbrook, Susan Pinker, Thomas Trumbull, Bernardo Seixas, Pablo Santurbano, Simon Columba, Jorge Espinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Inguanzo, Mikkel Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Ugni, Alexander Dunbauer, Omari Hickson, Fergal Cusson, Yevon Bodrenko, Hal Herzog, Don Ross, Jonathan Librant, Oslam Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, David Sloan Wilson, Yassila Dez Araújo, Ethan Solon, Romain Roach, Dmitry Grigoriev, Diego Londoño Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Elman, João Linhares, Lida Cosmides, Saima Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa, my producers, Izar Webb, Jim Frank, Lucas Tafini, Akian Gilligan, Sérgio Quadriano, Luís Caetano, Tom Van Egdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardas France, and Nirban Balachandran, and my executive producers, Michel Rugieski, Rosie, James Pratt, and Matthew Lavender. Thank you for all.